Hi-C is a technique that looks at the spatial conformation of our genome. Why do we need to look at the spatial conformation of our genome? Well, it turns out that location matters not only for school and work, it also matters for our genes. And who we hang out and spend time with matters not only for people, but also for genes. Because of that, there are various lab tests that detect if genes are located at their correct locations. And Hi-C is yet one of those techniques. Hello, welcome to Dr. XY's channel, where you're going to hear all things fascinating about genetics. Hi-C is a type of chromatin conformation technology. It looks at the spatial interaction between different regions of our genome. If we imagine our genome like a forest with many trees standing next to each other, their beautiful leaves and branches would crisscross with their own and others. And if you and I go for a walk in this forest, and with some superpower, we capture photos of all the trees from all the angles. Using these photos, we should be able to answer questions like, do maple trees tend to grow next to pine trees? This is a question of 1-to-1. We should also be able to answer what kinds of trees tend to grow next to, say, maple trees. This is a question of 1-to-many. And we can even answer the question, do any kind of trees tend to grow next to any kind of trees? This is a question of many-to-many. And regardless of how we ask questions and what we're looking at, one thing we can know for sure is that the branches and leaves that grow next to each other on the same tree would always tend to show up in the same photo. And just like that, chromatin conformation capture asks the question of spatial interaction between different regions of our genome. And depending on how complicated the question is it a 1-to-1 or 1-to-many, many-to-many, or all-to-all, it can be then called 3C, 4C, 5C, or even Hi-C. Hi-C answers the question of all-to-all. Regardless of that, we know for sure the genes and genomic regions that are next to each other on the same chromosome, they would always have high tendency to get captured together by this technique. So if we illustrate this with a very simple genome, here is what we can see. Say this is a genome with just 2 chromosomes. We have chromosome A with genes 1, 2, 3, 4. And we have chromosome B with genes 5, 6, 7, and 8. And if we plot these 2 chromosomes with 8 genes on X and Y axes, and we say every time when we capture the interactions between 2 locations, we're going to plot a red dot in the graph. And because the genomic regions, the genes that stand next to each other would always have a high chance of being captured together, the red dots are expected to be crowded along the diagonal line in the center of the graph. What if there is an abnormality? Say there's a translocation between chromosome A and chromosome B. If we graph their interaction, the remaining normal chromosomes that are not involved in the translocation are going to give us the dots that are on the diagonal line. Now, because one of the chromosome A swapped a segment with chromosome B, and the breakpoints are located between genes 2 and 3 on A and 5 and 6 on B. The genomic regions and genes near these breakpoints are not going to have a high chance to be captured together. This would show up as clusters of red dots outside of the diagonal line. So using Hi-C, we can detect structural rearrangement like this, and we can also spot where the breakpoints are. There is a wide range of application for Hi-C and other types of chromatin conformation capture techniques. What I'm talking about today is only one of them, which is the detection of chromosomal structural rearrangement. However, unlike most of the other applications that are not yet mature for patient care, the detection of structural rearrangement of our chromosomes is entering clinical labs. And that's super exciting, and which is why I'm talking about it today. So if you enjoyed today's video and would like to learn more about lab genetic tests that can help with patient care, Click like, share, and subscribe, and be sure to check out my previous videos on duplex sequencing and optic genome mapping. Thank you so much for visiting. This is Dr. XY, and I will see you next time.